All right, today we're talking about something truly groundbreaking, a new tool that's basically a universal translator for biology. It lets scientists read and compare some of the most complex maps ever created, the maps of our very own cells. So just imagine for a second, what if you had a Google Maps, but for the human body? Something that let you navigate the trillions of cells inside our tissues and zoom all the way in on any single one to see exactly what it's up to. I know, it sounds like something straight out of science fiction, right? Well, it's actually starting to become a reality. You see, this incredible field of technology is called spatial omics, and it already gives us these stunning, super high resolution maps of life itself. We can literally see exactly where every gene is firing up and where every single cell is sitting in relation to its neighbors. Okay, so we've got these beautiful, incredibly detailed maps. But what happens when you need to compare two of them? Let's say one map from a perfectly healthy brain and another from a brain with Alzheimer's. And this is where things get really, really messy. Trying to compare these two is like trying to overlay two maps of New York City, but one was drawn in 1890 by one artist, and the other was drawn last week by a totally different artist using a different scale. All the key info is there, sure, but getting it all to line up, it's next to impossible. I mean, look at all the variables. These biological samples have different sizes, different shapes. They're captured with different technologies. They come from individuals of different ages. There are just no obvious landmarks to match them up. And because of that, making a direct, meaningful comparison has been a massive roadblock for researchers. Until now. So, how do you solve a problem like this? Well, a team of researchers has developed a brilliant solution. It's a tool that acts like a biological GPS, creating a kind of common coordinate system for all of our cells. It's called CAST. CAST, which stands for Cross-Sample Alignment of Spatial Omics, is a seriously powerful deep learning AI. It really is that universal translator we've been talking about. It can look at two completely different biological maps, find the hidden patterns and similarities, and then perfectly align them, right down to the level of a single cell. And it works in three really elegant steps. First, the mark step. A neural network scans each map and finds common molecular landmarks. You can think of these like unique biological street signs. Then the stack step physically warps and stretches the maps to line up those landmarks. And finally, projection, where it merges all that data into one unified, multi-layered map that's finally ready for some real analysis. So what can you actually do with this biological GPS? Well, this is where it gets really exciting. Let's look at its first superpower, the ability to pinpoint the exact molecular changes that drive a disease. We'll use Alzheimer's as our example. So scientists took brain slices from healthy mice and from mice that had Alzheimer's-like symptoms. Now, just looking at them side by side like this, you can't really see the tiny, crucial molecular changes that are causing the disease. But by aligning them with CAST, they could run a special feature called delta analysis. The best way to think about it is like digitally subtracting the healthy map from the diseased map. What you're left with is basically a heat map showing only what's different, the hotspots of disease activity. Now, this is where it gets really, really cool. The analysis found two totally distinct molecular hotspots. Check out the chart. One cluster of genes, C1, was lighting up right where those toxic amyloid plaques were forming, and another cluster, C3, was active right around the p-tau tangles inside the cells. These are the two classic destructive signs of Alzheimer's, and for the first time, CAST showed exactly which genes were firing in those precise locations. But pinpointing disease is really just the beginning. The second superpower of CAST is that it can act like a search engine for biology, helping to build what's called a cellular atlas. Okay, imagine this. Let's say you're a researcher and you have a tiny, anonymous slice of brain tissue from a biopsy. How on earth could you figure out its exact origin from within the massive, complex landscape of the brain? Well, with CAST, you can take a molecular snapshot of that little piece and use it like a search query. CAST then scans a huge reference atlas that's made up of hundreds of different brain slices. And in this two-step search, it first narrows it down to the most likely locations, and then it does a super high-resolution alignment to find the single best match. It's literally like finding the exact spot for one puzzle piece in a 10,000-piece puzzle. It's incredible. And that ability to search for and place a sample perfectly within a bigger map brings us to CAST's final and, honestly, maybe its most revolutionary superpower, integrating completely different types of biological data into one single picture. Think of it like this. One technology, called StarMap, can measure gene activity. That's the cell's blueprint, which instructions are available. Another technology, RiboMap, measures which of those blueprints are actually being used to build proteins. That's the action. Until now, you could see one or you could see the other, but you could never see both at the same time in the same place. CAST completely changes the game. By aligning a StarMap blueprint and a RiboMap action map from adjacent tissue slices, it creates one single layered view. And that is a huge deal because for the first time ever, scientists can see both the genetic instructions and the resulting protein activity in every single cell in its exact location. So what's the big takeaway here? By combining these maps, scientists can now measure something called single-cell relative translation efficiency. In simpler terms, it's basically a score for how well a cell is turning its genetic blueprints into real action. This tells us which cells are working overtime and which ones are just kind of coasting, giving us this dynamic, incredible new picture of how our brains actually work. So why does all this matter so much? Because CAST is a revolutionary leap forward. It's that universal translator we've always needed in biology, letting us compare data from any lab, using any technology, even across different species. It lets us build that Google Maps of the human body, a searchable atlas where we can finally pinpoint the true origins of disease and get a much, much more complete picture of life itself. Look, CAST is way more than just a new tool. It's kicking off an entirely new era of biological exploration. For the very first time, we have a way to make all of our biological knowledge compatible. And that leaves us with one incredibly exciting question to think about. What are we going to discover when every map of life from every lab in the entire world can finally be read as one single coherent story?